consists of our co-leads. It consists of our co-leads for our project teams. We also have the outreach team, Callie and I. We have our marketing team, and we also have our leadership and training team. And I guess now we can move on to the introductory Mentimeter. If everyone could just join with the QR code on the slides, or you can go to the link attached to the, I can put it in the chat, the link as well.
Okay. If everyone has I will be going to the Mentimeter now. Oh, everyone's already done it. Okay. Okay. So our first question is, what is biomedical engineering to you? Do we have any responses there? Is it showing up on people's? Oh wait, hide results.
I have a quick question. Are the panelists supposed to do this Mentimeter thing, or is it just for attendees?
You can if you want to. Yeah, it's just an introductory activity, so we can all kind of understand a little bit more about what biomedical engineering means to us.
Okay, cool.
Okay, so we're getting a few words: design, medical, healthcare, just anything you can think of regarding biomedical engineering. It doesn't have to be right, but first thing that comes to your mind. Prosthetics, that's a good one. Innovation. Okay, so as these words are streaming in, biomedical engineering is basically applying principles and problem solving of engineering and applying those skills to biology and medicine. So as you can see, there's nanoparticles. It touches a variety of fields like implantable medical devices, pacemakers, artificial hep, stem cells. You can see helping people, that's a good one. So biomedical engineers basically use engineering and to improve healthcare problems and essentially help people. And if those are all of the, move on to the next question. So we just wanted to gauge how many of you guys are interested in pursuing research on an undergraduate level, on a graduate level, all three, even a PhD. We have some good results here. So hopefully by the end of this presentation, you guys have a better idea of the current research going on and also how to pursue graduate studies. So with that, we will continue the Google presentation and introduce our panelists for you guys today.
Yeah, so for tonight, we'll be having 4 panelists. If everyone wants to start, Joseph, I think you can start sharing your presentation. Yeah, perfect.
Okay, can everyone see my screen? Just
yep.
Yeah, perfect. Okay, good. Great. Okay, so hi everyone. So I'm Joseph, as Natasha and Callie said. So Callie is in my APS 106 class where I'm one of the instructors. So I was invited graciously to present my research. I hope you learned something. Much of my days is spent with ultrasound, and I'll talk a little bit about that. And I just wanted to say before I start that all of you who are here should be very happy that you're so interested in pursuing research, so early on in your career. I didn't even know what a peer-reviewed paper was in my first year. So the fact that all of you are here to learn, I think is a great start. Okay, so, okay. So the title of my presentation is Ultrasound Imaging from Hearts to Hearts on Chips. So let's go. Okay, so I just want to go over some key vocabulary for my talk. I don't know the background of many of you in the audience. Maybe my BME members also know, and MIE, I should say. But just some things to know when I talk. So I might say the words in vitro, which in this case mean outside of the body. You might be used to the words in vivo in like mice and rats and humans. Here, a lot of my work is in vitro, so it's outside of the body. In like petri dishes or microfluidic chips, or just smaller micro scale environments. I'll also be talking about a certain type of cell called cardiomyocytes, which are heart muscle cells. I'll also mention organoids, which are aggregates of heart stem cells, and they can recapitulate physiological hallmarks of organ level heart function. I'll be talking about microtissues, which are millimeter scale aggregates of heart stem cells that replicate physiological hallmarks of heart tissue. So not organ level function, but tissue level function. Because I'm doing ultrasound, I'll be talking about acoustic properties. So that's essentially the properties of a material in response to sound waves. I'll be talking about bulk mechanical properties, which are basically the physical properties of a material in response to the application of a force. And I'll be talking about hydrogels, which are gels essentially that are 98% composed of water, very similar to that of within your body. So in vitro cardiac models require new analytical tools. So there's a lot of cardiac models that exist currently to study diseases and evaluate different drugs within the body. So on the left, you have single cell models where they isolate single cells from a human being, and they look at the different ways in which it interacts with a variety of environments. Then we can look at organoids, which I talked about earlier, that are again micro scale aggregates of cells that beat and can give us indications about organ level function of a heart. We can look at microtissues, which are basically stem cells that form a tissue naturally that exhibit many different physiologically relevant properties of the heart. And we then can look at cells within gels and their reaction to their surrounding microenvironment. And so today in my talk, or in my lab, or in our lab, I should say, we study two main properties. And the first one is contractile properties. So that's how fast does a cell beat, or how fast does a certain model beat? How is its rhythm? How does it change? How strong does it beat? And also mechanical properties. So in particular, stiffness. So how stiff a tissue would be, or how stiff an organoid is, or how stiff a cell is. It's of great interest in our research. And so you might ask, well, why use ultrasound to study any of this? Isn't there stuff already that exists? And there's a bunch of different commercial systems that can measure contractility, for example, but they're expensive. They require specialized materials, specialized expertise, and they have to be used on particular substrates. And they give us like very high resolution, so they can resolve very small detail, and they can do that very fast, but they can't do both at the same time. They can either do one or the other. The other way is stiffness, and there's different techniques that can do that. But I want you to imagine, how can I determine how stiff something is if I can't touch it? Like, when you go to the grocery store and you pick out avocados for your avocado toast and you want to see if it's ripe, you'd have to feel it. But imagine that you're not able to do that. How can you even conceptualize that? And so ultrasound essentially fills that gap to allow us to measure stiffness and contractility of something without ever having to actually touch that something. And so that's kind of the beauty of where I love my research. So the beauty of what I see. And so ultrasound imaging, as many of you know, and hopefully many of you have been in a picture like this, starts when we look at babies in wombs. And essentially in these pictures, we look at frequencies that are very low, so less than 10 megahertz in frequency. And here, what I mean by frequency is the waves that are the waves that penetrate, the skin or penetrate a medium from an ultrasound transducer. And all of you have seen a transducer, so it's easy to conceptualize what that is. But now if I increase the frequency of that transducer, so if I increase the resolution of my system, I'm actually able to get to a point where I can resolve cells just as well as like a microscope or an optical image. And so that is kind of where we said, well, why don't we take this and apply it to all these micro scale in vitro cardiac models? And so we did. And the advantage here that we have is we have high temporal resolution. So we can measure hundreds of thousands of signals per second. So, within nanoseconds, we can have a single measurement. We're able to look at micron level resolution. So as you can see from this image here, this side by side of an optical image and ultrasound image, we can look at, these are single red blood cells from somebody's blood. We can look at them just as well as an optical microscope, and we can do it on any substrate. So we can put the sample on anything and are able to look at it. And so today I'm going to talk about two main applications of ultrasound imaging. There's 3 in my project, but I'm only going to talk about 2. And hopefully by the end, I convince you that ultrasound is one of the coolest imaging modalities there is. And you tell everyone you know that it's the best thing of all time. So I'm going to start on the first one, and we're looking at contractility of single cells, of cardiac organoids, and of microtissues. And so we're using ultrasound to look at how these things beat in time. And so what I want you to think of is kind of the fact of an ultrasound transducer over a cell. And here I've approximated a cell to a rectangular prism. It's easier to conceptualize. And as a cell contracts in one direction, what actually happens is that it changes in its Z direction. So I think many of the engineers right now are taking MIE 100. So you know about the different directions, X, Y, and Z, or I, J, K. So here what happens is as a cell contracts this way, it actually moves this way. And so that allows us then to measure the change in the cell height because of its contraction. Now, what that allows us to do is to in our setup, which is elicited here in the top, where we have this ultrasound probe, we have a well plate, and we have a microscope underneath, we can actually look at how a cell beats and how it changes in time due to a contraction. And we can measure that using ultrasound. Now you can imagine that this is powerful because we can add drugs to this system. We can look at disease cells, and we are able to look at the different contraction profiles of a single cell. And so that's one of the things I focused on in my project, is being able to do that. And on top of that, one of the things I wanted to pitch to you earlier is that we're able to do this very fast, thousands and hundreds of thousands of signals per second. And so what I did in this experiment is I took a cell that was beating, and I compared optical measurements, so like I showed you on the previous slide, to ultrasound. And I essentially had a cell beat, and I measured contraction with optical microscopy, so just a microscope, like stereotypically, and I also measured it with ultrasound, and I wanted to look at the profile as a function of 10 seconds. So I measured a cell beating for 10 seconds, and what I saw was that low frame rate optical imaging, in many cases that people have, cannot actually resolve a lot of the important parts of a contraction profile. So if you notice here, around 4.6 seconds, the optical measurement in black is actually missing where the peak contraction of the cell actually is. And ultrasound actually picks that up quite well, as you can see in red. And so that's powerful because it shows us this gap in being able to detect the contraction of multiple cells in a petri dish. And so for us, that's really cool. Now we can take this beyond single cells and look at the state currently in tissue engineering. One of the state of the arts is microtissues and organs on chips. And so 2 things that we looked at was also measuring how these tissues move in time, and basically how they displace. So how much do they actually move as a function of time, and can we measure that with ultrasound? And then we took these cardiac organoids, which I defined in my key vocabulary at the beginning as an aggregate of stem cells, and said, well, hey, what if I, put treat this organoid with a drug? Well, how will it change? How will its beat rate change? And so we did that, and we saw that ultrasound was actually able to pick up the changes in the beat rate in a cardiac organoid as we treat it with drugs. Now, this is also powerful because if we take drugs from, a sick kid who has a specific heart defect, and we're able to create a cardiac organoid and treat it with a certain drug and see how the beat rate changes, we're able to basically screen drugs for use for a variety of diseases. Now, this is not the only method, and it's one of many methods that can be used, but we think this is pretty cool and we want to study it. So this is what we did. So the next thing that I want to talk about, and I know I only have 20 minutes, is basically looking at cells and gels. Now, cells in your body are around this microenvironment of different things, right? You have tissues and you have all these different components in your body. And we said, well, what if we want to look at how cells react to a certain microenvironment? And can we measure different properties of these cells without ever touching them within that microenvironment? And so what we did is we looked at acoustic properties, which I defined earlier as being properties of a substance in response to sound waves, right? So not being able to touch it. And what I'm going to tell you at the beginning is that we can look at microstructure. So the way that cells organize themselves within our body or within a certain gel. We can look at physical properties like the density, so how dense a certain substance is. And we can look at bulk mechanical properties, so how stiff the entire gel is. And our setup is pretty simple. It's exactly like earlier where we have a transducer, we have a petri dish, and we have a hydrogel, which is a gel made up of 98% water, and a cell within that gel. And so if we have cells within a gel and they have ultrasound that is penetrating through that gel and hits a cell, for example, the cell can scatter the ultrasound, which I'm going to show in a second, but it can also absorb part of the ultrasound too. And what that looks like is something like this. And I'm just going to take my pointer off for a second. Ugh, don't do this to me. Hold on. I want to get rid of the arrow. There we go, perfect. So here in this animation, we have an ultrasound beam passing in this Z direction. And every single speck you see on that screen can be considered a cell, okay? And what happens is as the wave passes through that entire region, in this case 20 millimeters, what you actually see is this scattering from different parts of the actual area. And that scattering is exactly what happens in our body when ultrasound penetrates through like our skin. Our cells scatter waves that are returned to the surface. If you notice, what happens is the signals actually go back to the surface because our transducer is up here. And when we detect the waves that are scattered back, we can actually figure out, how big these cells are. Where are they relative? What is the concentration of them? And that is actually really interesting, and it allows us to think of a variety of things and in science and in biophysics. And so people have done this in really cool ways, and I'm going to show some cool stuff too, where if I don't have cells in the top in an ultrasound image, it's just completely black. We don't see anything. And as soon as I add cells, we see like these bright spots that come from cells scattering the ultrasound waves that I just showed. Now you can do this again in a B mode image is what it's called, which is exactly what you saw in that baby earlier. And here we just have a gel and we filled it with different concentrations of cells. And we want to see how the image changes as a function of cell concentration. And what you see is as I increase the cell concentration, it gets brighter and brighter. And that's exactly what I want you to take away from that. And so we did this in our lab at high frequency. So all of the stuff I showed you before was at lower frequencies. We want to be able to do this in really, really, really tiny gels, so like micron level, hundreds of micron level gels, or even, millimeter gels. So we took 3 different thicknesses of gels. We loaded them with umbilical cord perivascular cells. That's irrelevant what type of cell it is, but just loss of cells. And we wanted to look at the measured acoustic properties of them. So we wanted to see, how fast does sound move? So that's the speed of sound. How much do we reflect ultrasound? How much ultrasound is transmitted through and how much is reflected? That's the acoustic impedance. How dense is the medium? So, is it, since hydrogels are 98% water, are we just going to get the density of water? And how much is the ultrasound wave attenuated? Now, that is the key one here, and you'll see why in the results. Attenuation just refers to the decrease in ultrasound amplitude because of scattering. So I showed you what scattering was, and like decrease in amplitude literally just means the signal is this much, but then when I add cells, the signals is that much. And that is really reflective of organization. And so I'm going to show the results. It might be overwhelming at first. But what we saw is that relatively across the board, Speed of sound stays the same across different concentrations of cells. Acoustic impedance relatively also stays the same. Density stays really, really close to water. But acoustic attenuation shows us that as we increase the concentration, we can actually measure the changes in cell concentration in a particular gel. And now that's really cool. Why? Because it allows us to now look at biomaterials that we implant in people, and be able to say, how many cells are actually needed to, properly recapitulate a tissue for a human being. And biomaterial implantation is a huge component of regenerative medicine. So I think in order to finish up on time, I just want to say a few things and summarize my research and just, the key points. So high frequency ultrasound is amazing. I love it very much, but it's also can non-invasively and non-destructively measure functional and mechanical properties of single cells, microtissues, organoids, and biomaterial-based therapies. And we can probe length scales from millimeters down to micro scale, so things you don't even think of, like so small. And right now we want to move into doing measuring using elastography, which is where we can measure elastic properties of single cells and measure how stiff they are across an entire cell. And maybe that's something you'll all learn in like a third-year mechanical engineering course if you ever get the chance to take one. And I didn't want to end just on research. I kind of wanted to talk to you all about things I wish I knew in my undergrad as a way to switch. One of the things I want all of you to think about is finding mentors to help you out throughout your entire career. I don't think anyone gets anywhere alone. I think it's very important. When I was in my undergrad, I wish I knew that I should apply to everything I possibly could, because if I'm never gonna, be successful, if I never submit anything or apply anywhere, I think that failure is the best teacher. I think any of my other panelists in research know that we fail every single day, and I think that it's the best way to learn, especially in research. You have to be okay with failing once in a while, and by once in a while, I mean 99% of the time. I think you have to learn to write well. I think something I never learned in my undergrad, or I took for granted, was the ability to write well, because all I do now is write papers and grants and scholarship applications, and learning to write is probably one of the most, if not the most, skill I think you can learn in your undergrad. So please, please, please do it. Spend more times doing things you never regret. I gave up a lot of things in my undergrad to be where I am now, and I wish I didn't. I have never regretted getting 8 hours of sleep. I have never regretted doing something I like. So I think you should definitely spend more times doing things you never regret. I think that I wish I knew consistency is better than motivation. I was motivated to do lots of things in my undergrad. I wish I was just more consistent. And lastly, you should celebrate, or second lastly, I should say, you should celebrate your wins. Sometimes the way that we think we do is never as good as the way we actually did, and we constantly underestimate ourselves. So I think that, celebrate the things that you do well. And lastly, and the thing I probably live by the most, is lift as you climb and always pay it forward. I have never got anywhere by myself, and every time somebody asks me for help, I will do everything I can to help them. So thank you for your time. I have to acknowledge a variety of people that none of you know, and I won't have to acknowledge them, but a variety of funding sources. And I hope all of you enjoyed the presentation. And if you have any questions, feel free, but also you can email me anytime for anything you need whatsoever. Thanks.
Thank you for that amazing presentation. I will now open it up if there's any questions, or anyone has any questions they can put in the chat. But if not, we will go on to Stefan now.
I have a question for you, Joseph, actually. I really enjoyed your presentation.
Thanks.
I was going to ask, there's one slide where you have I think it's like those kind of like waves coming off of like the cells as opposed to the Z-plane, I think it was. Is that simulated data, or is that real data?
Oh, you mean the animation of the
there was like
this one.
Yeah,
I simulated.
Simulated, nice. You made the simulation?
Yeah, I made the simulation in MATLAB.
Nice, very nice. It's very pretty to look at. I like it.
Oh yeah, is it? Oh, good. I'm glad you enjoyed it.
By the end, it gets kind of chaotic, but it's pretty in a different way.
Yeah. Well, yeah. I'm not going to say anything, but yes.
Awesome.
Anyway, thanks, Stefan. Yeah.
Okay. I will share my screen now. Let me see. this. Okay, what am I doing? Okay, I think I got it. Share screen like that. Yeah, okay. Oh, wait, hold on. Sorry, guys, I'm a bit technically challenged here. Okay, I think I got it. Share screen.
Okay, we could answer a question in the meantime until you get set up. Oh.
Is this it? Am I sharing the right screen? What do you guys see right
Yeah,
now?
yeah, I can see your presenter view. Oh, sorry, not the presenter view, like the regular view, like investigating the role of ECM
Okay,
in
cool,
mediating.
cool.
Yeah, that's okay.
Okay, I think there was a question for you, Joseph, though, before I begin.
Okay, sure. So Ethan is asking, what are my thoughts on a life sci health sci undergrad transitioning into biomed eng program? So what I'll say is there's no one way to any particular like endpoint. So U of T is actually really great about that. Like I know a lot of people in BME who don't have engineering undergrads, they have related discipline undergrads and they transition into it. So I think Ethan, like you could totally transition into a biomed eng grad program pretty easily. I think you just my feedback is try and get experience in like engineering research before you apply, because in some ways you'll just have that one thing against you that you don't have a eng undergrad, which could be like I guess get like negated if you have research experience. And then Darsh is also asking. So Darsh, that's a great question. So there's actually another imaging technique that I also did in my undergrad called photoacoustics, which is the use of using lasers and ultrasound together to measure properties of single cells. If you look up, his name is, I'll put it in the chat, but his name is Michael Kolios. He actually teaches at Ryerson. He uses photoacoustics to look at single cells. And that's exactly, he uses lasers, just like you said, he uses lasers for cell measurements. And so I can put that in the chat or message you privately about that to give you more information about that. Okay, if nothing else, I'll let Stefan go.
Okay, very nice. So I would like to thank everyone for joining in today. The title of my talk is Investigating the Role of the Extracellular Matrix in Mediating Nanoparticle Permeation. But before I share my research, I'll provide a brief overview of my journey to graduate school and hopefully provide some useful pointers towards getting into research during undergrad. And because you're at this event, I'm assuming you're already involved in research or you're trying to get involved. And I knew when I was an undergrad, I wish that I had more advice, like how Joseph gave at the end of his presentation. So hopefully this can be a little bit helpful to you to get started in research. So a little bit of background and disclaimer. Everything I say is my perspective and thoughts, and I encourage you to reach out to other students, like the people in this panel, and also beyond in order to hear different perspectives. And the purpose of this slide over here is just to give you some background on who I am and to illustrate a few important points. So I did my undergrad at McMaster University in the Life Sciences program, and I got involved in some cancer biology research. And today I'm in a different program in a slightly different field. I'm doing my PhD in biomedical engineering in cancer nanomedicine. So the first point I want to make here is that you can change your discipline. You can do your undergrad in one program and do grad school or undergraduate research in a completely different program or department. So just because you're in chem engineering or biomedical engineering or whatever it is, that doesn't mean you can't do something in neuroscience or AI or whatever it is. I know someone who did their undergrad in psych and are now doing their PhD in biomedical engineering. My second point here is that the department you're under doesn't always mean much at face value. So even though I'm in this biomedical engineering PhD, there are people in my lab who are also in chemistry and they do the same work. And the final point I want to make here is that you can begin a PhD either directly out of undergrad, as in a direct entry PhD, or out of master's. So you can complete a master's and then do a PhD, and each option has their benefits. And so when looking to get involved in research, I think the program you're in is less important. Like your undergrad program is less important than your work ethic and personality. And hopefully you can build some projects and experiences throughout your undergrad, which can help to demonstrate and kind of substantiate your work ethic. This will help you when you're applying to labs, both in undergrad and afterwards. And so you may be wondering, how do I get involved in research? So I'm going to describe this process kind of linearly, but it's usually not this clean in reality. So the first thing you want to do is, this is you over here, you're at U of T, you're having so much fun, ha ha ha. And then you want to get involved in research. So you have to find out what you want to research. And, if you have a personal thing that you're interested in, such as cancer biology, then you just can jump into that. But otherwise, you can look into the pages of different researchers at your institute or in other departments, or even at completely different institutions. Like, let's say you want to go to the States, maybe you look at Harvard or something. Once you know what you are interested in personally, you have to find a supervisor who does this type of research. So here we have a supervisor. And in research, the head of a research group is called a principal investigator, or a PI for short. And this head of the research group, you want to look into the research papers that they have published. And one way you can do that is using a website called Google Scholar. It's like Google, but just for research articles. Once you have an idea of the work that they kind of do, you can send out like a personalized email to them. And it's important to keep it short and to the point. It's a good idea to describe what your personal interests are and how they align with what the lab does. If you have relevant research experience and if your GPA is strong, then I would suggest you mention it in the email to the PI. I think it will make you more competitive. It's also a good idea to attach your CV, which is kind of like a resume, because the PI is probably going to ask for it eventually. So just be upfront and proactive about that. It will save them time and save you time. If you don't have strong grades or any research experience, that's completely okay. Maybe you just don't mention it at this point, but I will say that eventually it may come up during an interview or if the PI asks you for your transcripts. This is probably more important at the undergrad level if you're applying for like certain scholarships, or at the graduate level if you're applying for scholarships. Once you send off your email, you're kind of in this waiting stage. So you have to wait until you get a reply. If you hear back and it's positive, you're probably going to get an interview, and then you have a chance to go into the lab. This is great. If you get a rejection email, this is okay. It probably happens to most people. I don't know the statistics. I don't know if these statistics even exist, but just know that rejections are tough, but you have to think about why it was a rejection. It could be completely, has nothing to do with you. Sometimes you reach out to a lab when they're not looking to take in students. Maybe they just hired some people, so they don't have space in the lab. It could also be because of you. Maybe your interview skills or your application wasn't strong or your email wasn't clear. All of these things are things that you can ask the PI about, and then it can help you to move forward. Just like Joseph said, we're going to make mistakes, but it's up to us to kind of take the responsibility to look at what we can do going forward to improve. I would also recommend you apply to a bunch of labs, and this will open up your chances to get into more different places, and also gives you the opportunity to pick a lab which you think really suits you best. But I would caution against sending kind of boilerplate cookie cutter emails. I would have a personalized email, do your research on what the lab is actually doing. So it's a nice, tight, well-constructed email. If you have these copy-paste superficial emails, a PI probably wouldn't like that. And so if you do want to get experience, you can volunteer in the lab. And that means they don't have to pay, the PI doesn't have to pay to hire you. You can see if you can do a co-op, or you can do maybe a course-based research. So some sort of thesis or a research practicum course. This might depend on the program you're in, how available these options are. But I know this was something I could do at McMaster. And of course, you may want to be competitive to get into a lab. You might want to have experience, which brings me to my next point. And this is kind of this meme over here, which is, one of the greatest challenges is you want to get into something, but you feel like you need experience. But if you don't have experience, then how are you supposed to start? It's like this paradox. And so one kind of way to ease into research is these kind of summer research programs. And the point of this slide is to show you that there are a bunch of opportunities for easing into research. Maybe take a picture of this slide or do a screenshot. The idea here is not to go through all of these programs, but just to know that there are a lot of opportunities which do exist. And even if you're interested in these, take a screenshot. And some of these dates have already passed, like these two last ones in red, the deadline have already passed. And the point I want to make here is that you want to start early when looking for your kind of like next steps. These applications take a lot of time to do sometimes. They require reference letters, which you have to give supervisors time to write for you, or maybe a professor you took a course with. And you want to keep an eye out for opportunities which might come out in the future. So even this was due February of this year, that doesn't mean you can't apply next year for next February, let's say. This link right here at the bottom, oh, and double check all the information on this slide because it might not be 100% perfect. And Sunnybrook still has some unopened programs. That was another important point. And the other thing was, look at this link over here. It's a shortened link from shortened URL from U of T that has a bunch of different resources which you can look into. Another thing is, in research, in order to do the work, a supervisor has to pay for you unless you're volunteering. So that means you have to have some sort of funding. However, if you are doing undergraduate research and you come with your own funding through like an NSERC Undergraduate Student Research Award, USRA, it's much more likely that a supervisor will accept you. So if you can come with these awards, an NSERC, USRA, or another award, it's much more likely that a supervisor will hire you because they don't need to worry about finding money to pay you. If you're in your last year of undergrad and you're applying to research programs, it's even good to think about this if you're in early years and you're interested in research at the graduate level. Here are a bunch of scholarships you can look into. You can apply out of your final year of undergrad if you want to do masters for CGSM, or actually you can apply for CGSM and PhD as well. OGS, PGS, all these other ones. And so now I'll pivot into the research I do today and just quickly grab some water. So I'm going to talk about a little bit about my research, and I realize now it might be a little bit too technical, so please feel free to ask me questions. The title of this part is Investigating the Role of the Extracellular Matrix, or ECM for short, in mediating nanoparticle permeation. And so currently, systemic drugs are used to treat cancer patients, and they're administered intravenously, so through the vein, and travel throughout the whole body. But because these drugs are systemic, they kill rapidly dividing cells that are both cancerous, like the tumor here, as well as healthy cells. And nanoparticles are an attractive vehicle for delivering both imaging and therapeutic agents, as they tend to accumulate in tumors. However, after these nanoparticles are injected, they face some barriers before they can actually enter the cancer cells to kill them. And on the left here, we have a blood vessel which is lined with these endothelial cells. The two primary barriers which nanoparticles face when reaching cancer cells are these endothelial cells surrounding the blood vessel, as well as this extracellular matrix, this ECM. So in the first stage of getting to the cancer cells, the nanoparticles transport through endothelial cells, and then they permeate through the extracellular matrix. However, not all of these blood vessels in the tumor are alike. In fact, only a specific subset of these endothelial cells which line blood vessels are responsible for transporting nanoparticles. And these nanoparticle transporting endothelial cells are called NTECs for short, as in the title. So over here we have a 3D image taken with a light sheet microscope, which is just a type of microscope that allows us to take 3D images. And it shows vessels in green and these nanoparticles in red. And in box E over here, we see that there are several nanoparticles in red surrounding this blood vessel, which has these nanotransporting endothelial cells. But in this vessel here, which is green, it's just a normal blood vessel. There's not a lot of these NTECs. So as a result, there's not a lot of nanoparticle transporting happening. But surrounding these blood vessels, there is this extracellular matrix sometimes. And this extracellular matrix can restrict nanoparticles from permeating out of the endothelial cells that line the blood vessels. And so in here, we see a slice of a tumor blood vessel, which is lined with normal endothelial cells in red, and these nanoparticle transporting endothelial cells in green. And once nanoparticles transport through these endothelial cells, they can be blocked by this extracellular matrix, this kind of meshwork of proteins, which is what we see in blue, the ECM. And what we find in the field of cancer nanomedicine is that most nanoparticles accumulate in this extracellular matrix. In general, nanoparticles which are smaller are able to get further through this ECM matrix than larger nanoparticles. But this ECM is not everywhere. This extracellular matrix restricts nanoparticle permeation, but the density of this extracellular matrix and the distribution of the extracellular matrix varies throughout the tumor. So here we have blood vessels in red and extracellular matrix in green. What we see is the ECM surrounding healthy vessels is generally consistent in density and distribution along the blood vessels. But this ECM along tumor blood vessels is quite chaotic and varies quite considerably, with regions of excess ECM, which blocks nanoparticles from getting out of the blood vessel. Therefore, it can't reach the cancer cells. So there are these specific endothelial cells which transport nanoparticles, And the surrounding extracellular matrix, if it's present, can block subsequent nanoparticle permeation. Previous studies show that most nanoparticles get trapped in the ECM, but despite this, we also see that nanoparticles can get out of the blood vessels if they have a lot of NTEX. But we don't know right now what are the features of the extracellular matrix surrounding normal endothelial cells and nanoparticle transporting endothelial cells, and if this ECM density is different. So I asked the question, what are the physical features of the extracellular matrix surrounding blood vessels that mediate nanoparticle permeation? And to answer this, I have set my objective to be to determine the physical features of the ECM surrounding blood vessels that mediate nanoparticle permeation in tumors. And I'll answer this objective through 3 different aims. However, I only have enough time to go through my first aim here, which is to develop a method to quantify the physical features of the ECM surrounding blood vessels. And so this is my first aim. And the way I do this is I induce a tumor in a mouse, and I inject it with a dye that labels the blood vessels. Then I take the tumor out of the mouse, I cut it up into different slices, and I label it with these antibodies, which allow me to see the extracellular matrix. So that means I have labeled the blood vessels and the extracellular matrix. Using this microscope, I can create the 3D image of the tumor, and then I can analyze it. From my analysis, what I see are that the average vessel branch length is 20.6 microns, so very small. There's quite a big distribution. And there's, for this vessel network within the tumor, there's about 2.14 branches per vessel. And what's really interesting though is most of the vessels are surrounded with this ECM, about 70%. So that means around 70% of these vessels have this proteins around it, which block the nanoparticles from getting to the cancer cells. What we see here in this visualization is a blood vessel network with branches in red. And the endpoint of these branches are shown with white dots, which allow me to isolate each branch. And here in green, we have this extracellular matrix. And when we zoom in on it, we see that there are some vessels which are mostly covered with ECM. And there are other vessels which are virtually uncovered. And so as a result of aim 1, I will have a method to quantify the amount of ECM surrounding these blood vessels, as well as other features of the extracellular matrix and the blood vessels. I don't have time to go into the other details of my experiment, but I'll just show you what this can also look like when you have this extracellular matrix surrounding blood vessels. You can go through this whole meshwork and you can kind of see all these purple nanoparticles surrounded with these green extracellular matrix. And over here in this region where we have blood vessels that are surrounded with very little extracellular matrix, it's very red, not a lot of green, you find that there's a lot of nanoparticles. So maybe this does mean, after all, as expected, that if you have the ECM, it will block the nanoparticles from getting out of the blood vessel. The significance of this research is we can create a map of how these ECM features influence nanoparticles. And in the grand scheme of things, perhaps in humans, we'll be able to take a biopsy from a cancer patient and to screen the tumor to see if the tumor has a lot of extracellular matrix, which will block nanoparticles from getting to cancer cells. If they have a lot of this matrix which blocks the nanoparticles, perhaps they wouldn't be a good candidate for chemotherapies which use nanomedicine, for example. But of course, we're eons away from that, and I'm still in my second year of PhD, so I'll update you in a few years, and hopefully I can give you some more clear answers. I'd like to thank you guys for listening, as well as my supervisor, Dr. Warren Chan, and some people from my lab. And yeah, thank you.
Thank you, Stefan, for your presentation. I think we're running a bit behind on time, so we'll leave all the questions until the end, but feel free to pop them in the chat. And now we will move on to Julian.
Hi, yeah. Yeah, great presentation, Stefan. A bunch of questions for you, but I guess I'll have to keep it for the end. Okay, let me share my slide. Let's give me a second. Okay. This. Okay, share, and then here. Okay, you guys can see the slides? Yeah, okay, great. You can hear me fine. Cool, so hi everyone. My name is Julian Sénécal. And my presentation is on lipid nanoparticles for RNA vaccines and therapeutics. Shameless plug here. Follow me on Twitter and on LinkedIn. Happy to connect with you guys, and shoot me an email as well if you have any questions about research or undergrad in general. So a bit about me to start. So I am an NSci 2TO. I specialize in the biomedical engineering major. And really, when I first joined NSci, I was really interested in aerospace engineering. And so I joined a couple of clubs, and then I realized really aerospace wasn't for me. And it really was kind of like a happy accident, the way that I got into biomedical engineering. Warren Chan, who's Stefan's supervisor, was teaching MSc 160, like a first year material science course. And I really liked chemistry in high school, so I reached out to him. And I asked him if I could do research in his lab during my first year. And initially he said no, and he actually said no like 2 or 3 times. So, I kind of kept pushing. And then ultimately he was like, all right, I'm going to give this guy a shot. And that's really where I found my calling, and I really fell in love with biomedical engineering. Then in my second year, I did research at MIT in the Chung lab. I kind of wanted to pursue an interest of mine in neuroscience, and that lab had pioneered tissue clearing to look at brain and brain structure. And then after that experience, I wanted to get a feel of how it was like to work in the pharmaceutical industry. So I got a job in the manufacturing science department at BIOME. That was a great summer in California. I really enjoyed that experience, and to understand how it was to work in industry. And that's really where I realized that if I wanted to kind of, lead a group and do science either in industry or in academia, I had to do a PhD. And then in my 4th year, I did a thesis in the Sefton lab, again at U of T. And that's where I got interested in immune engineering, so how biomaterials can affect the immune system. And that really was, I guess, kind of became the topic of my PhD. So I'm a direct entry PhD now in my 2nd year. And I decided to stay in Toronto, and I'm very happy about that. So I joined the new lab of Omar F. Khan. So I'm actually his first PhD student. And so we're a small lab working together to develop non-viral gene therapies. And our lab is really interdisciplinary. So people have backgrounds in chemistry, immunology, genetics, and me, biomedical engineering. And our lab is really at the intersection of immunology, genetics, chemistry, and engineering. So and part of my research is to develop new mRNA vaccines. So you've probably heard of mRNA vaccines because of the pandemics. I'm just going to give you a short overview of how they actually work. So mRNA vaccines are actually composed of 2 components, right? There is the mRNA, and there are lipid nanoparticles. And lipid nanoparticles are essential for the delivery of mRNA to cells, as well as stabilizing the mRNA, because mRNA is a very unstable molecule that needs to be protected. So once these lipid nanoparticles are injected into the muscle, they're taken up by cells. And inside the cell, they're actually trapped in endosomes, which are vesicles inside your cells. And the lipid nanoparticle essentially acquires a positive charge inside the endosome that allows to break the endosome and then release the mRNA cargo inside the cell. And if you remember your biology course from high school, DNA makes RNA, makes protein. So the mRNA inside the cell gets translated into proteins, which are these pink balls here, and then the proteins are secreted by the cell. So in the case of the COVID vaccine, the mRNA-coded protein is the spike protein of SARS-CoV-2. But our RNA is way more than just mRNA. There are a bunch of different types of RNAs that have different functions. So RNA is really a software drug that can do many things, such as gene inhibition with small interfering RNA or antisense oligonucleotides, gene expression with messenger RNA, and as well as gene editing with CRISPR that won the Nobel Prize recently. So an example of approved drugs, so Onpattro and Comirnaty, both utilize lipid nanoparticles, and there are really a plethora of pharmaceutical companies trying to develop these RNA therapies. I just want to highlight Deep Genomics and Providence Therapeutics that are based in Toronto. So what are lipid nanoparticles? So most RNA therapeutics require lipid nanoparticles, which if RNA is the software, the lipid nanoparticle is the hardware to make the therapy work. And as I said, they're essential for the therapeutic to work, to stabilize the RNA, to avoid it from being degraded inside the cell, as well as to deliver the RNA to cells, because RNA in itself cannot penetrate cells because it's negatively charged and the cell is also negatively charged. So that's why you have to use a lipid nanoparticle. So lipid nanoparticles are composed of 4 different lipids. Really, the most important one here is the ionizable lipid. But you can see that really the design of these lipid nanoparticles was inspired by living cells that also incorporate phospholipid and cholesterol in their cell membrane. And the mRNA is designed by first designing the DNA for the appropriate protein sequence, and then transcribing that RNA in vitro to make mRNA. And then the lipid components and the mRNA are rapidly mixed in a microfluidic channel to make lipid nanoparticles. So lipid nanoparticles are really cool, and you can do a bunch of things, such as organ-specific delivery. So if you just tune the lipids, if you change the composition of the nanoparticles, some of them go to the lung, some of them go to the spleen, and some of them go to the liver, which is pretty amazing for different therapeutics, say that target the lung or the liver, and to avoid off-target effects. As well, lipid nanoparticles are vaccine adjuvant. So that means that they stimulate the immune response. So if you look at this graph here, because the green dots are injection of an antigen with the lipid nanoparticles, and that results in high antibody titers. But when you remove one of the component of the lipid nanoparticle, the ionizable lipid, you don't get an immune response, you don't get antibodies. And that's why lipid nanoparticles work really well for the COVID vaccines, because they stimulate that immune response that's required to get an antibody response to actually go and destroy the virus. However, these current nanoparticles are really good for vaccines, but they're not that great for therapeutics, where you don't want an immune response. So the unmet need is that we need new lipid nanoparticles with lower reactogenicity. So that means less side effects and less toxic, that are not immunogenic, that don't induce an immune response, for really to fulfill the promise of RNA therapeutics, such as protein replacement therapies or the treatment of inflammatory and autoimmune diseases. So if you just look at this graph here, essentially current lipid nanoparticles are the blue and purple lines. So you have high mRNA expression as well as high inflammation. And the goal is to keep that high mRNA expression, but to have a low inflammation. So how do we do that? Well, so for me in my thesis, it's really to kind of dissect the anatomy of the ionizable lipid to develop new ionizable lipid or new lipid nanoparticles with non-immunogenic properties. So if you look at this lipid that's currently used in Moderna's vaccine, it comprises a RNA binding region with a hydrogen bonding group and an ionizable group, as well as a biodegradable ester linker and carbon chains to allow for nanoparticle formation by hydrophobic self-assembly. And in the lab, I actually designed new lipids. using a medicinal chemistry approach. Then I test these lipids in mice to determine the transfection efficiency and their immune responses. So I designed for intramuscular delivery of mRNA, low dose of lipid, high mRNA expression, low reactogenicity, low immunogenicity. And the design space are, or at least the parameters that I considered when I designed these lipids are hydrogen bonding, the ionization profile, the biodegradability, the lipophilicity, nanoparticle assembly, surface charge, nanoparticle size, and so on. And the way that I actually test these nanoparticles is to formulate these different lipids with different types of mRNA that can tell me the transfection efficiency. So I look at how much the organs glow, as well as the immune responses, so looking at the phenotype of cells and the inflammatory cytokines and the antibodies in blood and the lymph nodes. So I will also get you kind of excited about the future of these programmable drugs and RNA, because it's a field that's really hot right now, and pretty much all the pharmaceutical company are looking at this, and there's a lot of research in that area that some of you might be interested in. So particularly a future that I think is really near is precision medicine of RNA therapies. So say you have a patient with a disease like multiple sclerosis, you could profile the genome of that patient to identify exactly the genes that are problematic, and then you can design an RNA payload to fix those genes, put it in the right nanoparticle for the application, and then administer it to the patient. And I think we're actually really close to this future. And active areas of research right now, beyond mine. So obviously I work on developing non-immunogenic nanoparticles, but also groups like the Chan lab where Stefan works are interested in looking at the nanoparticle identity in vivo. So looking at how the proteins that absorb to the particles actually affect where they go in the body. Another challenge is to encapsulate large and complex payloads, such as CRISPR RNA, delivery to specific cell types to avoid off-target effects, or delivery to the tumors and not other organs to avoid side effects. Nanoparticles with fewer components are also being researched, because right now they have 4 lipids, and it'd be great to have a particle with only 1 or 2 lipid or polymers. Also, for the computer scientists in the room, there's a huge push for AI and machine learning assisted drug discovery in that space for both lipids and RNA. Storage conditions are a big challenge, and I'm sure you've heard of the cold chain problems with the COVID vaccines, because they have to be stored on -80, as well as there's been preliminary data that shows that manufacturing process also affects the efficacy of lipids. So that's an area of interest, of research more on the manufacturing side. And lastly, something that I didn't really touch about today is RNA design. And there's a lot of research going into different types of RNA design, such as self-replicating RNA or circular RNAs that can be used for better stability, lower immunogenicity, and higher expression. So on that, I'd like to thank you for inviting me to talk today and acknowledge my funding sources. And I'm happy to take any questions perhaps at the end of this, this talk. Thank you.
Thank you for that presentation, Julian. It was really interesting. We do have one question in the chat, but I think we'll leave it till after Kimberly's presentation. Kimberly, if you want to go ahead to pull up your presentation, feel free.
Okay, can everyone see my screen? Oh, okay, great. Hi, everyone. So I'm Kimberly, and my project is on the effects of osteocyte mechanostimulation on the suppression of prostate cancer bone metastasis. So first, I'll just briefly talk about my background. I did my undergrad at McGill in chemical engineering. This program has a big emphasis on research, and there are a lot of research opportunities in that program. And I graduated last year in 2021. So here is our logo, and here are views from our building, where I spent a lot of time and classes and also studying and doing experiments. So my first research experience was actually at U of T in the MIE department. I worked under Professor Edmund Young, studying tumor angiogenesis on a chip. And this was also under the IBBME undergraduate summer research program, which I did after my second year. And currently I'm a first year master's student in the cellular biomechanics lab under Professor Lee Den Yu. So our lab does a lot of fundamental bone research. We study bone mechanobiology. So basically it's the response of osteocytes to a number of mechanical stimuli, which I'll get into in a bit. And we also study the fundamental cellular mechanisms that involve osteocyte mechanosensitivity for treatments involving a number of bone diseases, such as osteoporosis. We also have developed microfluidic co-culture systems for studying osteocyte and osteoclast signaling. And more recently, we've kind of transitioned into studying bone metastasis in breast and prostate cancers. So I guess the main motivation behind my project is that a number of cancers have a tendency to metastasize to bone. So breast cancers, kidney cancers, and lung cancers, and prostate cancers. So bone metastasis will occur in about 65 to 75% of advanced prostate cancer patients. And it has a very low survival rate of 6% over 5 years, with a median survival period of 24 months. And about 20% of the annual deaths associated with metastatic bone disease is from prostate cancer. And patients can suffer from a number of various complications because the bone remodeling is disrupted, and it can lead to severe pain and skeletal related events such as bone fractures. And currently there isn't a cure for metastatic bone disease. A number of treatments like chemotherapies and biphosphonates can reduce the size of the cancers, but eventually these cancers will develop resistance to treatment. And yeah. So a brief introduction on bone remodeling. The bone is a very dynamic tissue. It is constantly being resorbed and formed at discrete sites. And basically the old bone tissue is being replaced with new bone tissue. And the cells that are in the bone are osteoclasts, which are responsible for bone resorption. These are multinucleated cells derived from monocytes. Osteoblasts are these cuboidal cells that are responsible for bone formation. And osteocytes are these dendritic cells that are mechanosensitive. So bone remodeling begins when osteoclasts are activated, and they basically resorb the bone matrix. And then macrophages go to the resorption site. Then osteoblasts are eventually recruited to the bone resorption site. And once they form the bone matrix, they will eventually become osteocytes, or they can also become bone lining cells, which are inactive osteocytes. So osteocytes are the most abundant cell in the bone. They've gotten a lot of attention over the last 25 years because of their mechanosensitive properties. They can respond to a number of mechanical stimuli like a strain, fluid flow, and hydrostatic pressure. And they have been found to regulate a number of cells in their environment, such as osteoblasts, osteoclasts, endothelial cells, and even immune cells. And the mechanical stimulation of osteocytes can modulate a number of processes like osteoclastogenesis, which is the resorption of bone, and osteogenic differentiation, which is when osteoblast precursors are matured or differentiated into mature osteoblasts. So what happens in prostate cancer bone metastasis is that it goes through a vicious cycle, which is when the tumors migrate to the bone and they secrete a number of factors that can activate bone cells. And in turn, these bone cells will secrete factors that promote tumor growth. The reason why bone is such a favorable site is because the bone will have all of these growth factors that will promote the invasion and proliferation of prostate cancers. And prostate cancers will form, will basically form osteoblastic lesions. And this results in enhanced bone growth, but of poor quality. So the new bone that grows will not have the, I guess, correct mechanical properties that normal or healthy bone will have. And this leads to the skeletal related events like bone fractures and such. So my project basically investigates how mechanically stimulating osteocytes can regulate or suppress prostate cancer bone metastasis. So the mechanical stimuli that I'm using is oscillatory fluid flow. And I'm investigating this regulation or suppression of prostate cancer bone metastasis through direct cell signaling, which is interactions between the osteocytes and tumor cells, through indirect cell signaling between osteoblasts, osteocytes, and cancer. So basically we use osteocytes, mature osteoblasts, to look at how whether the mechanically stimulating osteocytes could have an impact on osteoblasts and in turn have an impact on the growth of cancer. And finally, through studying real-time interactions on a previously developed microfluidic chip. And this here, I'll be studying the extravasation when tumors migrate through a blood vessel, and how endothelial cells could also be regulated or regulate prostate cancer bone metastasis. So for our cell signaling studies, we use conditioned media. So basically we take factors secreted from mechanically stimulated or static osteocytes and also mature osteoblasts. And here is just a picture of osteocytes after undergoing oscillatory fluid flow with a peak shear stress of 1 Pascal over the course of 2 hours. And below, we take mature osteoblasts after 2 weeks of differentiation. And the red is just a stain to see the calcified matrix deposit, which shows that the cells have differentiated. And then basically we take the medium from these cells, we culture prostate cancer cells in this medium and measure the various measure outcome variables such as invasion, migration distance, cell adhesion, or viability. So for some of my future work, I'm currently doing my cell signaling experiments, but I'll be doing microfluidic chip studies with endothelial cells. So here, the cancer cells are seated in a microfluidic lumen or blood vessel, and the extravasation or the migration of the cancer to the separate channel containing osteocytes is observed over a few days. And this chip allows for the mechanical loading of osteocytes in the microfluidic device itself. Another direction that I'm not sure how this would integrate into my thesis would be studies using immune cells and more specifically macrophages. So immune cells also play a role in bone metastasis because they interact with both tumor cells and osteocytes. So we are thinking of looking into how tumor associate or how the mechanical loading of osteocytes could potentially suppress the pro-metastatic effects of immune cells. And that concludes my presentation. Thank you for listening.
Thanks, Kimberly, for that great presentation. I think we can move on to the general questions. So we have a question from Edith in the chat. I believe this question was addressed to Julian. So how did EngSci prepare you for your graduate studies?
Yeah, sure. Happy to answer that in more detail. Yeah, I'm sure there are probably a couple of EngScis in the audience, and the engineering science has a reputation at U of T for being a very difficult and challenging program, mostly because of like its structure, right? So it starts with like 2 years of general engineering, which are incredibly difficult, and then the 2 last years where you specialize with in the option, and I specialize in BME, and I did much better in the 2 last years of my degree than in the first 2 years, mostly because I enjoyed my courses more. But that said, I think that going through this very difficult and interdisciplinary curriculum, I'm sure the others can also say the same thing from their undergraduate program. It just really it teaches you how to do research. Doing research is not necessarily like it's not about how smart you are, but it's how hard you work and how creative you are. And I think it's in that sense there perhaps EngSci is kind of a double-edged sword. If you focus too much on just getting top grades and working very, very hard, that doesn't necessarily mean that you'll become a good researcher. So I think that's why it's really important to not only supplement essentially what the courses that you take with with research and actual hands-on experience to develop your critical thinking, your research mind, and reading papers. And I think that's really what EngSci did the most for me, is to allow me to get access to these opportunities. And I think that's the biggest piece of advice I would tell you, is maximize your opportunities in undergrad, and it'll just life will do the rest.
Great,
Yeah,
thank
and
you.
I guess.
Sorry. There was a question from Rubena about the research grants such as NSERC, and I believe that was for Stefan. If you'd like to expand on it, but I think you also put a note in the chat. So if you'd like to expand on it, the floor is yours. But I think we do have some questions in the chat now.
I'll just say quickly, so I actually didn't get a USRA when I applied in undergrad, but I do know people who did get the USRA. So like OGS, NSERC, USRA, Vanier, all these kind of whatever applications you can do, there's a few things to keep in mind in the reality of it. You need to have references. So those are oftentimes supervisors, but if you don't have a supervisor in advance, it can be challenging to get good references. In which case, you can talk to professors who you've done well in their class. This kind of speaks to the bigger point of, always try to think about what you need, like what are the components of any successful application, and like what are required to complete an application. So, but there are always ways to get creative to like find a solution, even when it might not be obvious. So even if you don't have research experience in a lab, you can talk to professors, you can go to their office hours to build a rapport with them. And that is like one way which you can get a reference letter. Another one is if you're part of a club or an initiative outside of your school, that's another person who might be able to be a reference. Just be mindful, sometimes it's good to have academic references. So, there's a balance between who you might want to ask. In terms of when you actually apply for the award, like I said, I didn't get it. Maybe some of the panelists did get the USRA and they can speak to it from firsthand experience. But when I was applying, I know that I was already in the lab which I applied for, and I didn't get it, but I co-developed my proposal with my supervisor. So part of the application process is you need to develop a proposal. However, there was another student which joined my lab only because they were awarded the USRA, but they previously were not part of my undergraduate lab. So they wouldn't have been able to have the chance to join my lab unless they had that funding source. But once they had it, they were retained in the lab. Yeah, but they didn't actually they weren't in the lab at the time of creating the application, but they did tell my former supervisor, hey, I'm going to apply for this NSERC USRA, just letting you know. I do have some questions about the research in the lab. Can you check over my proposal? Maybe I'll pass it along to other panelists if they have any other perspective on USRA or other scholarships.
I actually did receive a USRA. I'm not sure how much it's like changed because it was a few years ago. But I remember I just contacted, or I was already going to work with one of my former supervisors that coming summer. And I told him like, hey, I'm gonna apply for a USRA just to like have funding and stuff like that. I think I did not. I don't think I wrote the proposal. I think it was the graduate student that was supervising me who wrote it. And then my PI edited that as well. And yeah, you just have to fill out the forms with your PI, and then yeah, making sure that you submit it on time, depending on your department's deadlines, because I think each department can have like a different deadline for the USRA.
I don't have anything to add. I think mostly everyone covered it. I didn't get a USRA though. Well, like I don't even think I applied once in like my second year. And then my supervisor was like, forget it, I'll just fund you. So yeah, I didn't do that. But yeah, anyway.
Yeah, I might jump in for like, the summer research. I mean, for the undergrads that are starting to do summer research, it's well known that summer research doesn't pay very well, but that you shouldn't do research for free, and that if your PI really does not want to give you any money at all, like, and not even like $1,000 per month, you might be you can be in the position of saying, hey, I can't do this full time, right? And you shouldn't be expected to do research for free. And yeah, if you can, like, my God, I've had like so many experiences of, so yes, perhaps I can share like an anecdote of when I did research at MIT that summer. I actually got basically the funding before the prof knew that I was coming. Like I was in contact with like the postdoc, and I was like, hey, I want to do research with you. We wrote the application to like the NSI scholarship, sent it in, got the money, and then after that, call the prof and be like, hey, I have some money, can I come to your lab? And then he was like, sure, why not? So that might be a strategy that can work for some people.
Yeah, I did have one more question before I move on to the question in the chat. I'm not sure if any of the panelists did some sort of thesis or capstone project during their undergrad, but if any of you did, do you have any advice or sort of recommendations for how to go about doing a thesis in maybe fourth year?
I actually wrote my undergrad thesis. It was like the very start of COVID. I did have like our lab had like a Google Drive of like all the old thesis that previous undergrad students wrote. So maybe something, if your PI will let you, is to like ask them for maybe old thesis that previous students wrote, just so you have an idea of like how to structure it or what kind of formatting your PI is expecting from you.
I did my capstone at the same time as like I did research too. So I kind of made it not the same. I kind of wanted to learn something else. So for me, like, I was like, oh, I want to learn a different skill. And so I did my capstone something like completely different than what I was doing like, research wise. And I actually found that really helpful because it gave me insight into, other parts of research. I find that grad students like focus on what they know and like what they do. And like, we forget that there's like an infinite amount of other things that we could possibly be working on. So I think that exposing yourself to multiple things is really useful. I think that when you're in your undergrad, you should say yes to many things, as many things as you can. But I really want to emphasize Julian's point that do not do it for free, because like no one should be making you like work long hours and pay you nothing. That's kind of inhumane and not fair. So, but my main point is that I would say if you can learn new things, you should learn things. That I think that's good.
Yeah, I also want to build on that, and you also have to think about the next steps, right? So when you're doing your thesis, often like it's in fourth year, and often the PI, like the prof, also uses the thesis as like a trial run for a grad student. So try and perhaps join a lab that interests you and where perhaps you'll be able to learn new skills, but also that you might be interested in joining for grad school, or that a prof that has connections that could perhaps connect you with another prof at another school, by writing you a nice reference letter for grad school. So that's maybe like a more strategic take on it, but at least that's kind of how it worked for me.
What was the question again?
Do you have any advice or recommendations on how to approach a thesis or capstone project in undergrad?
Once you're already in the thesis, or to get into the thesis?
Kind of both. Kind of both.
Okay. So from my experience, I did a third-year research practicum, which is just like a semester of research. And then I stayed in that lab and I did a fourth-year thesis. So I guess in order to get into research, the biggest thing is honestly just take the initiative. It's scary and it's tough, but just do it. Send that email and the rest can follow. But it's like pretty much Joseph's like kind of equation thing. Like you actually have zero chance. You miss 100% of the shots you don't take. So you will never be able to be in a lab unless you try. You will never be able to get a job unless you try. Unless, like, I don't know, nepotism and someone in your family gets you a job or something, but don't count on it, And so I think just try, and it's easy to say, but harder to do. And then once you're actually in the thesis, I think the biggest thing is really, part of it's the technical skills, but the most important thing is I think more of developing your time management, learning how to write clearly and communicate clearly. And the reason why I say that is to build off of like what Julian was saying, it's really life after lab in undergrad, which I think is much more longer than your life in undergrad. Undergrad is 4, 5, 6 years, whatever, but life after that can be, so long. So if you think about the kind of the long game here, if you build these kind of core skills, these core competencies that will, that if you use your undergrad time and your thesis time to build these skills, it will pay you dividends throughout your whole career. The last thing I was going to say about the previous NSERC USRA question is, I'm pretty sure, but maybe someone can chime in or double check, you can actually apply to NSERC USRAs at other institutions. So even if you're at McMaster, like you can go to U of T and do an NSERC USRA.
Yeah.
And you can also use like I think the USRA, you might be able to also use it for like sick kids and like those other, I don't know if they're not private, but like maybe they are private, I don't know. But PIs at like institutions which are not universities necessarily. So that's like another point. And thank you, Kimberly. I think you said yes, right?
Yeah, because I had mine at U of T, but I went to McGill for my undergrad, so.
Oh, there's one more thing I want to add to in terms of piece of advice, is reach out to grad students before you reach out to the PI. Like that, like, profs are really, really busy and they might not answer your email, or they might forward it to grad students and then your email gets forgotten. But if you establish a contact initially with a grad student, like, we have, like most of us are like super nice and have the time to, take an hour out of your day to talk to you guys. And that could be a good strategy, because at the end of the day, you'll probably be working as an undergrad in close proximity to a grad student.
Yeah, definitely. Thank you, guys. That was really helpful. We do have one final question from Darsh. What's the most challenging thing you have faced in your graduate program, and what advice can you give us to avoid these challenges?
I can start. I think it's actually, I wouldn't ask this question. It's not about avoiding the challenges. For me, it's more about recognizing the challenges and overcoming it, because that's really where the growth happens. And the reason why I say that is because So, okay, undue challenges, like don't put yourself in a tough situation just for the sake of it, unless whatever that's your mentality. But I don't aspire to put myself in challenging situations, which I don't see a purpose to. But it's not, I really don't look at it as like, how do I avoid challenges, but rather, how can I choose challenges which are worthwhile, which will help me to grow? And the answer to that, I think, oftentimes is to be perseverant, keep going, try your best, ask other people for help. And the big picture of all of this is actually to provide help to other people. Everyone has things to do. We appreciate that you invited us here. And I also see, presenting today as an opportunity to give back. Like I said, I wish that more students in the graduate level gave these kind of presentations and had some guidance. And, everyone here has been very nice about giving out their emails. So, do reach out to us because we do want to help you. And, seek to challenge yourself, seek to help others, and seek help. And good luck.
Okay, I can also, I guess, go next. Yeah, so I agree that I don't think there's a graduate student alive that can say that they were not challenged during their degree. So I don't think it's about avoiding challenges, because like some people, there's this saying that there's was in my lab, which is like, you finish a PhD in 2 weeks because those are the only days that your experiments actually work, is like 2 weeks in a year. So it's just like things like that where like 90% of the time like things don't really go as well as you hope they do, and that's okay. So I don't think it's about them being challenging. I think the most challenging thing for me was exactly what Stefan just said. It's just being perseverant. I think that again, like I think everyone is dropping such tidbits of wisdom, but so mine is kind of focusing on what Julian said earlier, which is that just because you're good at memorizing stuff for school or doing well in courses does not mean you're going to be good at research. I know plenty of 4.0 GPAs who can't do an experiment to save their life. So I don't think that it actually means anything in terms of really anything. Yeah, it's helpful for awards and it's helpful for certain things, but in the grand scheme of it, like, does it predict success? I wouldn't argue yes. So what I want to say is I think undergrad prepares you to like deal with like, you don't do well on a test. But I think that being perseverant and learning and being curious and following, curiosity and trying things and asking people for help and like really just like reading and writing and like there's so many different responsibilities that you have, that managing them all and staying perseverant and motivated is incredibly difficult. And I think that is one of the most challenging things I have felt, because sometimes I just want to sleep, but it's really tough. So I would say being perseverant is one of the most challenging things, but I think it's about managing yourself and putting yourself first and being able to do that while also doing graduate research.
Yeah, okay. Yeah, if I can jump in. Yeah, I agree with everything that was just said. I think there's scientific, there's interpersonal challenge, and you need to tackle them and to be resilient in the face of them. But I think that for me, the biggest challenge, especially in my first year, was to change the beat, of like, especially when you're a direct entry PhD, you go straight from undergrad to a PhD, where like, in undergrad you work like 24/7, like, always like the way you study for tests kind of, and now you enter a PhD, and I was just kind of like always on all the time, and then got really tired doing that. And then that the challenging part was to kind of realize like, hey, a PhD is, it's a long game, right? It's like, it's a marathon. It's a 5-year, 6-year process. You got to take care of yourself. You need time to actually like step away from your research to think more and to plan and, to read. And that's not really something that you learn necessarily in undergrad, because you're always like doing assignments and homeworks and writing midterms. So that change of pace, it was definitely challenging. But really important, because I know a lot of you, especially like in undergrad, you might maybe you're suffocating right now with so much work, but know that when you're in when you're in grad school, sometimes you have a bit you need to learn how to breathe, essentially, because that's how you do good research.
Okay. If anyone has anything to add, or we move on to the next. We have 2 more questions. Sorry, apologize
I have
for
one thing
going
to
over
add,
the time limit.
Callie,
Oh,
if I can
go
add
ahead.
one thing. Yeah, sorry to interrupt. I also think that if all of you are choosing labs, like that's great. And I think that if you're thinking about people, it's important, but make sure you pick someone that like you like if you want to do research with them, or kind of find them that they're supportive. I think that so many students pick like this amazing lab at like Harvard, but they see their supervisor once a year and like they don't really talk to them. And as someone who wanted to go to the States, got in, but choose to stay at U of T, I think that it's important that you remember that prestige is not everything. And that like it's really about your day to day, because once you're at that place, the shine of a certain name brand school wears off, and what you're left with is the environment that you're around, the people that you're around, and the work that you do. So I would say like make sure that you find a place that supports you and that really cares about you, and a person like a supervisor or a PI, like Stefan mentioned, that cares about you. It's more important than you realize, and it will pay you dividends going forward. That's all I have to add. So sorry, go ahead.
Yes, 1,000 times. Yes, whatever he said.
Great, thank you. We have one more question. I believe it was for Joseph about the cardiac stiffness. Give the floor.
Sure. Okay. So essentially, I think Lara is asking, how do we stimulate the cells, which is, I guess a big transition from what we're talking about currently. Yeah, essentially we use like electrical stimulation to stimulate the beating of the cells when we were imaging them, because yeah, there's no nervous cells for the nervous system. So yes, I'll just leave it at that. But if you have more questions, I put my email in the chat. So if anyone has questions, feel free to ask.
Great. I believe that was all the questions. I think there's one, is any of your labs hiring at the moment? But I guess you can email them. So at this time, if there's no more questions, we'd like to wrap up this event. Thank you to all our graduate students for taking the time to speak with everyone and share your words of wisdom. And the recording will be made available if anyone wants to reference back and watch the recording. So thank you everyone for joining.
Yeah, thank you everyone for joining.
Thank you.
If the panelists could stay back for a few minutes, that would be lovely.
Thank you for having me.
Yeah, thank you everyone for being here.
Thanks everyone for coming.
Yeah, good luck, guys. Thank you.